Today I'll be presenting my poster presentation entitled Mutation Analysis of the K13 Full Gene, PFMDR1 gene, PFCRT markers that are present in Plasmodium falciparum isolates collected in Western Kenya in comparison to clinical phenotype. And in this regard, we're talking about the ring stage survival rates as well as the delayed parasite clearance rates. So recently we've had artemisinin resistance introduced or rather reported from Southeast Asia, and this is a concern to us and also worldwide. And so for this reason, we are looking at the genetics that could affect the clinical aspect, and basically looking at the PFMDR1 and PFCRT marker, we are seeing that the wild type to PFCRT, that's case 76, the wild type to codon 84, 86, the mutant side to 184, and also 1246, the wild type. We look at all this because we know the haplotype to PFMDR1 may be influencing the artemisinin resistance. And so we are able to also look at the percentage that is high as opposed to that could suggest there is artemisinin resistance. And looking at the mutations present in the propeller region, we are seeing that there are no mutations that are reported, like in the table shown. There are no mutations reported as seen in Southeast Asia. But in regards to the same region, we are looking at the several mutations that have been seen. But clearly, when we look at their significance, there is no significance and also correlation to artemisinin resistance. And also in the graph that shows the ring stage survival rates, we are reporting that the mean survival rates for RSA is 5.23, which is safer to say that most of our parasites are sensitive to artemisinin drug. But just to look at the parasites that have a longer survival rates, more than 10, and we are seeing the parasites of the same sort that are still surviving after drug has been given after 72 hours. So this is a concern, but still we are glad that the preliminary findings are encouraging and suggest that there is no artemisinin resistance yet reported in Kenya. But looking at the variations in the full gene, we are able to see that there is an increased repeat section in the amino acid position 137 to 142, which has been observed as opposed to the wild type 3D7 parasite. So we are seeing two parasites, 2% of our isolates had 8 repeats, and then 2% also had 7 repeats. So it is evidence that genetic determinants for higher survival rates for ACTs in Africa is different as compared to Southeast Asia. This is to let us know that K13 may not be the particular marker that could like elucidate the mutations present, but for sure we are seeing that there is longer survival rates and also delayed parasite clearance rates in our data. And preliminary phylogenetic analysis showed that there was no correlation between the genetics, looking at the full gene of the 13th chromosome, and also comparing with the survival rates of those isolates that had more than 10 survival rates. So among the isolates, we noticed that there is a mutation, K189 mutation, that had the highest frequency. But also, despite the fact it being non-synonymous, it is not significant to let us know that there is to be correlated to artemisinin resistance. Other unique mutations have also been seen and also reported in other parts of Africa, but still we have those cases of synonymous mutations which are not really adding to information as well. So the phenotype of these coding substitutions are unknown, but really at this point we require further characterization, and also more research should look at the molecular levels or the genetics to really elucidate what's going on, because K13 marker doesn't show us, but still we have those parasites that have longer survival rates. So since K13s have not been shown to be prevalent, we at this stage we are able to identify, we are able to advocate that there should be more molecular markers that should be critical in monitoring emergence and spread of artemisinin resistance. So that's the way forward, and we appreciate for where research has reached and all our funders. Thank you.